All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. And first of all, thank you to everyone who had sent good wishes or condolences for my younger brother's death. Thank you again. And let's start our discussion for today. What I want to do is this is the FDA and CDC um, XBB monovalent vaccine approval documentation. It happened on June 15th. However, someone sent it to me from our Substack and said that can you review how they were looking at the vaccine efficacy and the testing and the trialing that was done for authorization. So as we go over this data, I would request you to keep a couple of things in your mind to look in the data, and that is what is the duration of efficacy of the boosters? And secondly, what is the method for analyzing the efficacy for the future boosters that are now approved or authorized for 2023 fall and winter campaign? I think you'll find it interesting. I would give you another context here. A few days ago, I shared a study, and that study was about 800 times higher incidence of myocardial injury. And someone wrote this comment. I'm giving you this comment as a context. I looked at the study. N equals 777. Number of people participating, 777. Seriously? This is an absurdly small number of individuals included. This isn't even close to enough of a sample size to draw any meaningful conclusion. I did not expect much sense in this video, but it just goes to show why critical reading is so important in this day and age. So this is someone upset about 777 people included in a study to be a small size. So if you then contact FDA and CDC, they will say, hold my beer, I can do better. And now they would show you authorization based on either data in a few mice, 20 mice, for example, or a few people, and that few 20 to 50 people. So keep this in mind that interestingly, the scientific community, medical community, has reached a point where they dismiss or accept the study based on basically their liking. However, they will try to nitpick the problem wherever they can. So I wanted to see how the FDA and CDC, they care for the data and how much care do they have. So let's look at the data itself. So all the links are in the description. If there is some link that is not present, you can simply Google it. Here is where we'll start from. Vaccines and Related Biological Products Advisory Committee, June 15, 2023 meeting announcement. The basic idea of this meeting was that we had, if I can quickly draw as well, I love to draw and explain. So we had an original vaccine that was against the Wuhan or wild type virus, the original virus, SARS-CoV-2. Then we had, so this was a monovalent vaccine because there was just one mRNA against a spike protein of the wild type, or called WT. Then came the BA.4/5 time, remember? And what happened was WT vaccine mRNA was present in the dose, plus they added the more contemporary variants mRNA as well. And doses were set up in a way that half of the dose was mRNA for wild type, the other half was the mRNA for the newer type. And this was called bivalent. I still remember we talked about the bivalent, and one reason that bivalent was approved or authorized was that the committee doctors, the doctors in the committee, they said, and you can go back and listen to it, and I had done that talk at that time as well. They said they felt comfortable with the wild type mRNA to be present in the newer mRNA dose. So the idea of the science was the feeling comfort. And I still remember that I thought it's not about comfort, it is about previous one not working, and that is why we need the newer one. So why to keep the previous one there? And you would see today in the documents that I'll go over that the Pfizer, Novavax, and Moderna, I think one or two of them said that having the previous variant's vaccine causes immune imprinting, meaning it teaches our immune system to react to the previous variant. And so when the new variant will come in, because of that immune imprinting, the new variant will not have a good chance to be attacked, because the virus would try to produce antibodies against the the body will try to produce antibodies against the old variant. You would actually see the thought of immune imprinting presented here as a negative now to say we need to move on from the bivalent boosters. So this is science happening. That on one end, they're saying, well, we feel comfortable. And at that time, the thought of immune imprinting was present. Now they want to move away from bivalent. So all the arguments are in favor of a new monovalent. And these are good arguments that, hey, we don't want immune imprinting, so let's go to the monovalents. It's not a wrong argument. The same argument was applicable before, where I feel comfortable resulted in bivalent. And you can hear that that video where they were talking about we feel comfortable. Okay, so then now what is happening is that the XBB time, and this time is almost over, XBB 1.5 and XBB 1.6 point whatever, these variants are kind of escaped to a majority of this vaccine and even previous infection. So now they're saying, you know what, we should have a monovalent vaccine that only recognizes this variant and not the previous ones. And the reason for saying that is that there is immune imprinting that happens, which results in confusion of the immune system and not an efficient handling of the new variants. So sample size is its own issue. Monovalent to bivalent back to monovalent is another interesting journey you would see for the science community, and see how they tread away from science when needed and then come back, but they still continue to call that this is all science. Now, the links here that I'm going to go over, they are present at the end of this document. So if you go down here to the table, event material, all of these links are to various documents that I'm going to show you today, so you can find them here. So this is the FDA briefing document, Vaccine and Related Biological Product Advisory Committee meeting, June 15. This is a preparation document, so I'm going to leave this. And let's jump right into the documents presented by various vaccine companies. So this first one is the CDC itself. So CDC is saying we need a different booster. And here is a reason for asking for a monovalent booster. So let's very quickly skim through this one. First, they are saying over here, what is the coverage of vaccination? So if you see here, at least one dose, lesser than 2 years of age, 8.9%, greater than 65 of age, 95%. And at least one bivalent dose, 43.3% greater than 65. And then, so this is that data. Then they talk about bivalent vaccine efficacy, monovalent and bivalent. So we're going to look into that. So I'm just going to go directly to the efficacy. This is CDC presenting to the public, all of us, to say the vaccines aren't working. We need a monovalent vaccine. And that is the reason that they're putting this together. So here is how to look at this one. Instead of just trying to read the numbers, also look at this column where you would see these purple ones are bivalent vaccines, and this is within a 59 days time. So that is within 2 months, the efficacy. Then within 2 months to 194 months, efficacy reduction. And this efficacy is against hospitalization and critical. It is not about cases. And then if you see down here, it is for the 120 to 179. So 4 to 6 months. So by that time, efficacy pretty much just goes down. And if you see here, this is the unity line, or this line at the gray line at 0. If at any point you see the efficacy crossing this line, then that would mean efficacy does not exist. So for example, here they're saying, I'll just read one for our interest. This is the absolute vaccine efficacy of monovalent and bivalent booster doses against hospitalization and critical illness among immunocompetent adults aged 18 plus, September 2022 to May 2023. Let's look at this top chart. Hospitalization, unvaccinated as a reference, monovalent dose only. If you see here, 21% is the efficacy. You may have noticed that originally, when the pandemic started and the vaccine discussions started, the expected and asked or demanded efficacy by the agencies was more than 85% to 90% for severe cases. Here we are looking at monovalent, that is wild type, not the new monovalent. This is the discussion done to say we need to have the new monovalent. So the monovalent written here this time is the old one, 21%. Then if you look at booster of bivalent within 2 months, 62%. Then 2 to 4 months drop to 47%, and 4 to 6 months, 24%. The other thing that you would see here is that the side effects are not discussed. It is not discussed that if you have repeated boosters and if you are a healthy individual, what could be the cost benefit analysis. That is not here. Then I'm going to continue. This is the absolute vaccine efficacy of monovalent and bivalent against hospitalization among immunocompetent adult age 18 for BA4 and 5 variants. And once again, if you see here, monovalent has an efficacy of 25, bivalent within 2 months has efficacy of 61%, and then within 2 to 4 months, it drops down to 40%. Here, if you see a similar thing, this is for XBB, the previous monovalent and bivalent boosters, XBB. Monovalent, no efficacy. If you see, this is crossing the gray line. If you see here, -4 to 20%. So as soon as it crosses the unity, it is ineffective, or data is not significant, or it's a fluke. So monovalents are useless now in XBB time. And then bivalent, 51% within 2 months. And from 2 to 4 months, it drops down to 20%. Then they have another reporting system, IVW, investigating respiratory viruses, the acutely ill, in the acutely ill. That data, again, if you see here, this is very interesting. Look at this data. Absolute vaccine efficacy against COVID-19 hospitalization among immunocompetent adults aged 18 plus, September 8, 2020 to May 29, 2023. And look at the bivalent vaccine 120 days onwards, 4 months onwards, -6, -27 to 30. It crosses the unity. That means it is useless data. It means there is no efficacy. And it drops from 54 within 2 months to 34% between 2 to 4 months. And then after 4 months, no efficacy. Then if you see here as well, the bottom row set is XBB. And they're talking about booster, bivalent booster, 7 to 89 days, meaning within 3 months, and 90 to 179 days, 3 to 6 months. And if you see here, efficacy does not exist at all. So for me, that is interesting, that they're making a case to say, we need to have a new monovalent vaccine for the contemporary variant. And in that, making that argument, they're saying that, hey, these vaccines boosters are actually not doing anything. What is interesting for me is, did they ever come out and say these before June? Did they ever come out to say these are not working? Stop taking them. Because here, look, XBB, January 23 to May 24, 2023, January to May timeframe, before June this year, booster bivalent for XBB -8 to 53. That means it crosses unity, useless. Bivalent booster dose 90 to 117 days earlier, 3 to 6 months. Efficacy -8, -44 to 19, meaning useless data, nothing there. Alexander says, I am immunocompetent against COVID. Yes, you are, Alexander. So this is what surprises me. And of course, when I say this is what surprises me, then I get a lot of people's comments that you are naive. Fine, I am naive. But I still feel surprised that in June of 2023, our scientific community, our leaders of science, owners of science, are saying we need to have a monovalent booster because we have seen that the previous boosters do not work at all. And here is that data. Do you see this data? XBB. So anyways, this is CDC now trying to make a case to say, hey, we need to have this data. Then this is for pregnancy. Then there is data for EDUC encounters, etc. So there is more data here. Now I want to go to the companies who were asked to present their data to request for authorization of their monovalent vaccine. And what is that data? Let's see. Moderna. So I'm going to just skip over things. Moderna said, hey, we had an original vaccine against a wild type. Then we had a bivalent vaccine, wild type plus BA.4/5. And then we are unvaccinated individuals, and we do a comparison. And they create an absolute vaccine efficacy between unvaccinated to vaccinated, and relative vaccine efficacy between one type of vaccine to the other. And here are the vaccine efficacies. So if you see here, 4 doses, for example, look at this last row, more than 4 doses of the vaccine. Original vaccine, 27% efficacy. Moderna's bivalent vaccine, 46% efficacy. This is against hospitalization. So again, if you see here, relative efficacy 70%, 83%, 55%, and absolute here similar numbers. What is interesting, so then they talk about the new variant. So there is this BA1 versus BA5 and XBB versus XBB.1.1.6. So they talk about that. But then what is interesting is they would say preclinical data and then clinical data. And you would see that Pfizer probably is not going to even be able to offer that, and Novavax also not. So here, what are they talking about is cross neutralization at day 29. Day 29, within a month, following Omicron BA.4/5 bivalent booster. So this is the neutralization of BA.4/5 of the bivalent booster. And this is trying to have that neutralize the antibodies, and there neutralize the XBB. This is not in person's assessment. This is cross neutralization in lab. Then minimal antigenic differences, some more data that way. But then they continue on, and here is the preclinical data. 14 days mice after the booster dose. God forbid if this was the kind of data available on some other study coming from outside of these companies. The people would come out with pitchforks. As I read to you, that 777 individuals study was being mocked as a small study. And the presenter was mocked for not having critical reading skills. So here is FDA, CDC doing one up. And we have mice 14 days after the booster dose. And this is what the data is. Now, this monovalent here in Moderna's, this monovalent is the new monovalent that is against XBB. And if you see here, this is the bivalent, this is the monovalent, and this is the BA4 bivalent. And these are various variants. And again, they're saying that monovalent against XBB works better on XBB. So then that was the preclinical data, that is mice study. So if you see here, this is mice, this is mice, and so this is also mice. Then they go to clinical data, which is human beings, and look at the number here. Phase 2-3 randomized safety and immunogenicity study of XBB.1.5 containing booster in adults 18 plus. Randomized April 25 to 27, N equals 101 number, 1 N50 for monovalent, N51 for bivalent. This is the data. And it is not some hidden data that is sitting in some corner. This is the data that Moderna brought to the table. Is it Moderna? I think it is Moderna. They brought to the table saying, hey, we've done some mice studies, and here we've done some studies on 50 people. And based on that, we've given them this dose. And based on that, we feel that you should authorize our product. Nobody raised their hand and said, this sample size is very small. And now based on that sample size, here is the data analysis. And again, if you are interested in seeing what these doses are doing, then look at this. XBB.1.5 and XBB.1.16 neutralizing antibodies after 5th dose, 3rd booster of XBB containing vaccines in adults. So if you see here, these bars are the orange bar is the XBB efficacy against that, and then the greenish bar is bivalent vaccine and the efficacy against that. So again, clearly monovalent is showing efficacy against the XBB ones. Similarly, more data over here, but once again, 50 individuals. For me, what is interesting is 50 individuals data is the clinical data set presented by these companies, and we are okay with it. Our agencies are okay with it. So neutralizing antibodies, as you can see, monovalent XBB variant, XBB.1.5, XBB.1.6, 232, and the effects or efficacy for them. So anyways, this is that data. This is Moderna. Now let's look at Pfizer. Let's see what they do. Let me see immune imprint. So no, they don't. Then I think that is in Novavax. It will be very interesting to see that. So this is Pfizer. How does Pfizer put their communication forward? Pfizer is saying weekly proportion from 1 April to 20 of XBB is becoming bigger. Then they talk about the previous one. The previous monovalents and bivalents have failed. They didn't say failed, but efficacy has gone down. So look at this table that they're putting forth as an argument to say these are not working very well. So see here, absolute vaccine efficacy against hospitalization, monovalent, age 18 to 64 years here, monovalent 7 days after, efficacy 17%. Booster then 7 to 59 days, 2 months, 61%. Booster 2 months to 4 months, almost finished, 1%. It's almost touching that zero to become useless. And then, of course, then 4 months later, minus 24 to 43, gone. Similarly, time since mRNA, age 65 plus. And once again, if you see 24, 64, 51, and 27. So they are making a case to say these previous ones aren't working. Then they're saying in the fall and winter, there are going to be huge number of new variants. So we need to be ready. And then they also take some help from immunology. And they talk about the CD4 cells and CD8 cells. They say expand spike-specific CD4 and CD8 responses, recall spike-specific memory B cell that recognize the shared epitopes, results in superior variants. So they give you some data from there. Now here, this is neutralization assay. So boost neutralization activity against this bivalent. So that's that. But this is B cell, and they're talking about B cell memory cells, immune imprinting would occur. And so we want to have the correct vaccine variant so that the correct memory B cells are formed. If we make incorrect B cells and then we encounter the new variant, then the previous B cells will try to respond and we'll be in trouble. And they talk about more of those. I want to go to the data itself. So preclinical evaluation. This is vaccine authorization we're talking about. And of course, preclinical evaluation, to the top right, you see the cute friend of ours over here, mouse. Now, how many mice? Total 20, 10 mice per group. 2 groups, 10 mice per group. That is a preclinical data. So here is one mouse study. There is some more data from mouse. Again, from a data point of view, if you're curious, they are showing that in the mice, the monovalent latest booster is working, and that's what they're showing here. And it is interesting that the monovalent starts working more towards the newer variant, and the previous variants it does not, because they have escaped. Then they say, we are ready with the supply. So we will give XBB 1.5 monovalent in end of July, 1.16 in August, any other formulation in October. Then they say, and this is what really made me angry, then they say that, hey, 60% of the flu doses are distributed by end of September. We will combine our doses with them. And why this made me angry is that last year when these meetings were happening, and remember there was a signal about the strokes happening in some people, and the CDC's data showed that this stroke was occurring more in those who were taking a combined flu plus COVID boosters. And they simply said that because we see the data, CDC, but FDA didn't see it, then it is okay. And I presented at that time as well, the definition of the cases were different. So one agency looked at a different set of strokes versus the other agency. And because then the data did not match, they simply said, well, one agency saw the signal, other did not see it, so it's okay. And all they had to do was to say, take the two vaccines separately. And I know that their hesitation was that somebody who's there to get the vaccine, if you give them one vaccine and ask them to come back 3 weeks later, they would not come back. So these are quite reckless decisions, but they did them. So here, once again, Pfizer is saying, well, we'll combine this with the flu. Primary presentation will be single dose units, enabling greater access and efficiency. Now, here is a preclinical and clinical data. Did you actually see any clinical data in this presentation? Did you see any human data in here? You only saw the mice data here, this data. But they put the clinical anyways, because the clinical is for the previous boosters. But there's no clinical for the newer booster. But they said, hey, clinical and preclinical. So they right in front of you, just put the words in there and hope, or they actually are confident that you would just ignore it. So they just called it preclinical and clinical data support, a monovalent XBB adapted vaccine. So this is Pfizer. Now let's go to the last one for today, Novavax. Novavax came up with, of course, once again, hey, this is a vaccine, this is a vaccine, prototype and XBB1 pseudovirus neutralization response. So again, this is pseudovirus. Remember that you build a virus and you put the spikes of the XBB on it, and then you try to neutralize it in the lab. This is that data. And here, if you look at this data, the titers, this is prototype and this is XBB, and the empty ones are mRNA vaccines, and the colored ones are Novavax vaccines. So not only they are presenting their case, they're also kind of competing with the mRNA as well. So then you see this one, primary 2-dose series in mice. So neutralization in mice, primary vaccination with 2 doses of monovalent prototype, XBB 1.5 or XBB 1.6. So once again, these colored ones are Novavax, and this is pseudovirus plus mouse, and this is their efficacy. Or really not the efficacy, the neutralization of the antibody levels. Neutralization in mice, primary vaccination with 2 doses of monovalent or bivalent prototype plus XBB. So this is that data. But again, my point, if you would like, I can go over these data to make sense of them. But my point is, these are not human studies. These are also not very large studies. These are 10 mice here, 10 mouse there. And then they have another one, and that is booster dose data in mice and macaques. So they have monkeys here as well. So this is that data. This is Kyrie, who is scratching at the door. And so now this is the spike protein mutations. They talk about that. I believe in here they're going to talk about the imprinting as well. Then they talk about T cell responses. And remember that Novavax had said that we go towards cytotoxic T cells. So here they're talking about CD4 plus T cell responses in mice. And they're talking about various kind of interferons, etc. Here they're talking about the CD4 responses in rhesus macaque monkeys. Then data support. Then they say Novavax data supports a monovalent vaccine for 2023-24 season. This is it. So to be, and then there is FDA takes action and goes ahead and authorizes it. Individuals 5 years of age, individuals 6 months through 4 years, unvaccinated individuals, and so on. So they go ahead and they authorize that. Interesting thing is for me to see that the data is just. So what do you think? Do you think that it is actually fair that original vaccines were tested on 40,000 people or 30,000 people, and because that test was done, the whole platform and the vaccine technology has become verified, and so now if there is a new mRNA in there, it's just 10 mice are now sufficient to make sure that there are antibodies produced, and that's it. What do you think? Is this fair? Is this scientific? Or there should be more rigorous data? For example, I'll tell you my opinion. My opinion is that even if they were going to do this, they should have done it for the adults first, and then see if they need to go back to the youngers later on based on the results in adults. And even in the adults, then the recommendation should not have been everybody go get it, because there are really, we all have a lot of protection now from the vaccine or the infection or both. They should have said, here are the groups that may need it because their own immune system may not be competent enough to handle this. So anyways, that data, we've been saying that from the beginning, and they have not been listening from the beginning. So saying this would not change anything. But it is interesting for me to see how the variability in decision making occurs from feeling comfortable to 10 mice to in vitro to monkeys or to 50 individuals, and that is how the data is used. Skyfrog, welcome. Skyfrog says mice have rights too and lefts. Absolutely. So with this, thank you very much for chiming in and thank you very much for tuning in as well. Taking a vaccine or not taking a vaccine is absolutely your decision, between you, your family, your doctors. Nothing to do with me. You take it, more power to you. You don't take it, more power to you. For me to say it one way or the other is medical advice, and I'm not giving a medical advice. It is just interesting to see how the science has deviated from science, and wherever it goes, that is now called science. So thank you very much. Please like, subscribe, and share, and I'll see you tomorrow. Bye for now.